There are lakes at the bottom of the ocean, not metaphors, not geological formations that resemble lakes, actual lakes with shorelines, waves, and surfaces you can see, sitting miles underwater, completely separate from the ocean surrounding them. Scientists have found them, filmed them, and some of what lives beside them may be the closest thing on Earth to the conditions that started life itself. This is what they found down there. Most of what sits at the bottom of the ocean has never been seen by human eyes. The deep sea floor is not a place humans visit casually. The pressure at those depths would crush an unprotected human body in seconds. The darkness is total, and the cost of sending equipment down and retrieving it safely runs into the millions. For most of recorded history, the deep ocean was simply a blank, a place scientists knew existed but could not meaningfully access. That began to change in the latter half of the 20th century as remotely operated vehicles became reliable enough to be deployed at serious depth. ROVs don't need life support. They don't need pressurized cabins. A camera and a set of sensors strapped to a metal frame can descend to places no submarine could safely go, transmitting footage back to researchers sitting in a warm room on the surface, watching a monitor. For the first time, the deep sea floor became something scientists could actually observe rather than just theorize about. What they expected to find mostly was more of the same: sand, rock, sediment. The abyssal plain is vast and by most measures monotonous, a desert that covers more of the Earth's surface than all the continents combined, largely featureless, largely empty. When the research vessel Ocean Explorer headed into the northern Gulf of Aqaba in 2020, the team on board was not anticipating anything unusual. The mission was routine, running sonar over the seafloor, collecting depth data, filling in the gaps in charts that were already mostly complete. The Red Sea is not a deep ocean. It averages only about 490 meters in depth, shallow by oceanic standards, and the stretch of seafloor beneath the Gulf of Aqaba had been surveyed before. There was no scientific reason to expect anything remarkable. The sonar was running, the data was coming in, and the ship was moving steadily through water that had been considered well understood for decades. But the sonar data didn't make sense. The readings coming back from the seafloor showed depressions, pockets in the rock that registered differently from the sediment around them, as though something was sitting inside them that wasn't sand and wasn't stone. The team ran the scan again. The anomaly stayed. Whatever was in those depressions was returning a signal that the equipment had no category for. But even that wasn't the part that stopped the team. Anomalous sonar readings happen. Equipment picks up gas pockets, unusual rock formations, pockets of sediment with different density. There are explanations for most strange readings, and experienced survey teams develop a tolerance for data that initially looks wrong. The Red Sea floor is geologically active. It sits on a spreading ridge, the same tectonic boundary that is slowly pulling Africa and Arabia apart, and unusual formations are not unheard of. The team noted the readings and deployed the ROV. But what the ROV camera transmitted back to the surface had no geological explanation that anyone on board could immediately reach for. The image showed a surface, not rock, not sediment, a surface that moved slowly, almost imperceptibly, but visibly, in the way a body of liquid moves when something disturbs it slightly. A surface that reflected the ROV's lights back in a way that solid material doesn't. The researchers on board had collectively spent decades studying the deep ocean. Not one of them had seen anything like what was on that monitor. But the strangest part wasn't the image, it was the location. The readings placed these formations at 1,770 meters, deep, but not extraordinarily deep by oceanic standards. And the GPS coordinates put them close. Uncomfortably close to shore, in water that ships pass through regularly, in a region that had been surveyed before. Whatever was down there had been there the whole time. 4 pools. That's what they found. Not one strange anomaly that could be explained away as a sensor glitch or a trick of the camera. 4 distinct bodies of liquid sitting on the seafloor of the Red Sea, each with slightly different chemistry, each with a perfectly defined shoreline, each completely separate from the ocean surrounding them. The team named them the Neom Brine Pools, after the futuristic development project being built on the Saudi coastline nearby. And what made the discovery genuinely unsettling wasn't just what the pools were. It was where they were, just 1.2 miles from shore, the closest brine pools to human civilization ever recorded. The sonar returned data that simply didn't match what a seafloor was supposed to look like. There were depressions. Inside those depressions were readings that suggested separate, distinct bodies of liquid. The equipment was checked. The diagnostics came back clean. The readings stayed. When the ROV went down and the cameras switched on, what appeared on the monitor was something almost no one in oceanography had seen with their own eyes. A shoreline. Gentle waves moving in slow motion along a visible edge, a surface that reflected the ROV's lights back like a dark mirror. The water above it was clear. The water inside it looked wrong, thicker, heavier, almost syrupy. You could see exactly where one ended and the other began, as though someone had drawn a line across the sea floor. What the Neom discovery confirmed wasn't just that brine pools exist. Scientists had known that for decades in other parts of the world. What it confirmed was that they can exist anywhere the geological conditions allow, including places nobody thought to look. If 4 major brine pools had been sitting undiscovered near a populated coastline until 2020, the question that followed was immediate and uncomfortable. How many more are down there? But before anything else, there is a question that nobody who sees brine pool footage can get past. The camera is already underwater. So what is that surface? What is that visible line sitting on the seafloor, with waves moving along it and a shoreline around it, behaving exactly like the surface of a lake? The answer involves a property of salt that most people have never had reason to think about, and once you understand it, the lake stops looking impossible and starts looking inevitable. The first thing most people ask when they see footage of a brine pool for the first time is whether the camera is malfunctioning. The image on screen shows what appears to be the surface of a lake, rippling slightly, reflecting light, with a visible shoreline and what looks like a fog or haze hanging just above it. And yet, the camera is underwater. The submersible is already submerged. Whatever that surface is, it is not the boundary between water and air. It is the boundary between 2 different bodies of water. The question that follows is immediate. How does that work? Water is water. Pour one body of water into another, and they mix. That is the most basic behavior of the substance. It finds its own level. It disperses. It equalizes. Every childhood experiment with a glass and a faucet confirms it. And yet, here, at the bottom of the Red Sea, 2 bodies of water are sitting next to each other with a clearly defined boundary between them, behaving as though separated by an invisible wall. Scientists who study brine pools described the boundary as one of the most visually striking phenomena in deep-sea research. The ROV footage shows it with startling clarity. On one side, normal seawater, clear, dark, the kind of water that fills every ocean on the planet. On the other side, something denser and darker, almost syrupy in the way it moves, with a surface so distinct that you can see the exact line where one ends and the other begins. The two bodies of water are in contact, and yet they are not mixing. To understand why, you have to think about what salt actually does to water, not just the taste of it, but the physics of it at a molecular level, in quantities far beyond anything found in a kitchen or a swimming pool. But salt, on its own, doesn't explain everything. Add salt to water, and yes, the water becomes denser. That part is straightforward, but the density difference between normal seawater and even moderately saline water is not dramatic enough to produce a visible, stable surface line. Stir a glass of water with a pinch of salt, and nothing separates. The salt distributes. The water remains a single, uniform body. For a brine pool to maintain a boundary sharp enough to see on camera, sharp enough that you can watch an animal cross it, the salt concentration has to be extreme. Not slightly elevated, extreme. But even extreme salinity alone doesn't fully account for the stability of the boundary over time. Salt is soluble. Given enough time, even a very dense body of salty water should gradually lose material to the less salty water above it. Particles migrating upward, the concentration slowly evening out, the boundary blurring and eventually disappearing. The lake should be draining itself, and yet brine pools are not temporary features. Some of the pools in the Gulf of Mexico appear to have been stable for thousands of years. The Neon pools were sitting on the Red Sea floor, fully formed, when the ROV found them in 2020. They were not new, they were not dissolving. But the question that took scientists longest to answer wasn't why the boundary exists. It was why it stays. Why the brine doesn't simply diffuse upward into the ocean over time, the way any dissolved substance eventually distributes through any body of liquid it contacts. Something is actively maintaining that line, not a physical barrier, not a container, but a process. And that process turns out to involve two forces working against each other in a specific and unusual way that depends on a property of water that most people never think about. The reason brine stays put comes down to density, and density alone. Dissolve enough salt into a volume of water and it becomes heavier than the water around it. Not slightly heavier, significantly heavier. Brine pool water is dense enough that a submersible moving downward through the ocean will physically bounce off its surface the way a ball bounces off a trampoline. The vehicle doesn't pass through. The brine simply refuses to give way. That density is what gives brine pools their lake-like appearance. The brine sinks into the natural depressions of the seafloor and stays there, held in place not by any container, but by its own weight. No current that sweeps across the seafloor can disturb it, and the seawater above it has no mechanism to pull it apart, so the brine just collects, pooling in the low points of the ocean floor the way water fills a bowl. The boundary itself is maintained by a process called double diffusive convection. Heat travels through water roughly 100 times faster than salt does. So when the warm, salty brine sits beneath cooler, less salty seawater, the heat begins to move upward while the salt lags behind. The water directly above the brine surface gets warmer and rises away from the pool. The water directly below the surface gets slightly cooler and sinks back down into it. The result is two layers that actively resist mixing, a thermal current pulling upward on one side, a density gradient pulling downward on the other. That process maintains the sharp surface line that makes brine pools look, against all logic, like a lake sitting at the bottom of the sea. Some of these pools are small, a few meters across, barely wider than a room. Others stretch for miles. In the Gulf of Mexico, certain brine basins are large enough that the brine flows over the edges of underwater ridges, creating waterfalls, rivers of dense liquid cascading down the seafloor in slow motion, in complete darkness, miles below the surface. The physics is straightforward once you understand it, but knowing how it works doesn't make it look any less impossible. So the physics holds. The brine is dense enough to stay put, and the process of maintaining the boundary has been running, in some cases, for thousands of years. But that raises the next question, the one the physics answer doesn't touch at all. Where did the salt come from? Getting it to the bottom of the Red Sea required something that happened millions of years before any ROV ever descended to find it. Once you understand that brine pools exist because dense, hypersaline water sinks and collects in seafloor depressions, the next question seems straightforward. Where does the salt come from? The ocean is salty, but not nearly salty enough to produce brine on its own. Normal seawater at 3.5% salinity cannot become brine simply by sitting still. Something has to concentrate the salt far beyond what the ocean naturally contains, and that process doesn't happen quickly, and it doesn't happen anywhere. Brine pools have been confirmed in only 3 regions on Earth. That is not because scientists haven't looked elsewhere. Deep sea survey work has covered ocean floors in every part of the world. The limitation is geological. Brine pools require a very specific set of conditions to form, and those conditions exist in only a small number of places. Finding out what those conditions are means going back not thousands of years, but millions, to periods when the ocean itself looked completely different from how it looks today. The Red Sea is one of the youngest major bodies of water on the planet. It began forming roughly 30 million years ago, when the African and Arabian tectonic plates started pulling apart, and it is still widening today at a rate of about 2 centimeters per year. During its formation, the sea went through cycles of connection and isolation, periods when it was linked to the broader Indian Ocean, and periods when that connection closed and the water trapped inside had nowhere to go. What happened during those closed periods is what makes the Red Sea floor so geologically unusual today. The Gulf of Mexico has its own separate history, older and deeper, shaped by different tectonic forces over a longer timescale. The Mediterranean went through perhaps the most dramatic transformation of the 3, a period called the Mycenaean salinity crisis, roughly 5.6 million years ago, when the connection between the Mediterranean and the Atlantic fully closed and the entire sea nearly evaporated over the course of perhaps 1,000 years, leaving behind a vast salt desert kilometers below what is now the seafloor of southern Europe and North Africa. But knowing where brine pools exist doesn't immediately explain how the salt got there in the concentrations required. The ocean is old. It has been cycling water for billions of years, and dissolved minerals have been accumulating in it for just as long. But the background salinity of the global ocean, around 3.5%, is nowhere near what brine pools require. Getting from 3.5% to 30%, or in some Red Sea pools, to concentrations many times beyond that, is not a matter of degree. It is a different category of chemistry entirely, and it cannot happen through the normal processes of oceanic circulation. But the geological history of each brine pool region holds a specific event, or in most cases, a cycle of events that produced salt in quantities the surrounding ocean could never replicate. The key in every case is isolation. A body of water cut off from the global ocean behaves completely differently from a body of water that is connected to it. It cannot replenish itself. Whatever is inside it is all there is, and under the right conditions, the right climate, the right temperatures, the right surface area, an isolated sea will simply evaporate, leaving behind every mineral it ever carried, dissolved in solution. But the evaporation is only the beginning. The salt left behind doesn't stay on the surface where it formed. Over millions of years, sediment buries it. Tectonic movement compresses it, shifts it, forces it deeper into the crust. The original salt deposit, already enormous after multiple cycles of flooding and drying, gets folded into the geology of the region, locked beneath layer after layer of material that accumulated on top of it over timescales too long to easily picture. And then, slowly, the water finds it again. The salt that fills these pools is ancient. In the Red Sea, the deposits feeding modern brine pools began forming during the Miocene epoch, roughly 20 million years ago, when the Red Sea basin repeatedly flooded and then dried out as tectonic plates shifted and the connection to the Indian Ocean opened and closed. Each time the sea evaporated, the salt stayed behind. Each time it flooded again, sediment washed over the salt and buried it. That cycle repeated, layer over layer, until enormous deposits of evaporite sat locked beneath the modern seafloor, not as a single seam, but as a geological stack, salt and sediment alternating like the rings of a tree. In the Gulf of Mexico, the mechanism is different, but the origin is the same. There, salt domes, some over a mile tall, have been squeezed upward by tectonic pressure from below. As they rise, they fracture the sediment above them. Seawater seeps down through those fractures, dissolves the salt, and the resulting brine flows back out onto the seafloor through the gaps. The brine doesn't collect gradually over centuries in the Gulf of Mexico. It leaches out through cracks like oil from a well, pooling wherever the seafloor is lowest. What both processes share is the same starting condition, a body of water that was once cut off from the global ocean. The Red Sea, the Gulf of Mexico, and the Mediterranean are the only places on Earth where brine pools have been confirmed to exist, and all three of them were sealed basins at some point in their geological past. That isolation is what allowed the salt to accumulate in the first place. Without it, evaporation would have been replaced by new water flowing in, and the salt would never have built to the concentrations needed. The lakes sitting at the bottom of the ocean today are the direct result of something that happened to the surface of those oceans millions of years ago. So the salt has a source. The pools have a geological history stretching back millions of years, built from evaporated seas and buried deposits and the slow pressure of tectonic movement. But knowing where the brine came from doesn't tell you what it does. The footage from the ROV shows animals approaching the pool's edge, some of them crossing it. And what happens after that crossing is not gradual, and it is not survivable. Whatever is in that water kills everything that enters it. The question is how, and the answer is faster than almost anyone expects. The shoreline of a brine pool looks, in footage, almost peaceful. The surface moves in slow, gentle ripples. The edge of the pool is clear. You can see exactly where the brine begins, a visible line against the sediment. Animals move through the water nearby, some of them passing very close to the boundary without appearing to notice it or be troubled by it. The scene has a quietness to it, a stillness that is unusual in deep-sea footage, where hydrothermal vents hiss and sediment drifts and currents pull at everything in frame. That stillness is deceptive. The boundary of a brine pool is among the most abrupt biological dividing lines found anywhere on Earth. Not a gradient, not a zone of transition, but an edge. On one side of that edge, marine animals live and move and feed as they do everywhere else in the deep ocean. On the other side, they die, not slowly, not over hours or days, but within seconds of contact. The footage from multiple research expeditions shows the same sequence. An animal approaches the surface, crosses it, and almost immediately its movement becomes wrong, erratic, uncontrolled. The fluid coordination of a living creature replaced by the mechanical, helpless motion of something that has lost control of its own body. What makes the brine pool boundary so lethal is not a single property of the water. It is several properties operating together, each one independently capable of killing, all of them present at once. The chemistry inside a brine pool is, from a biological standpoint, comprehensively hostile, not in the way that cold water is hostile or deep pressure is hostile, both of which organisms have adapted to across evolutionary time. The brine pool is hostile in a way that leaves no margin, and adaptation takes time that the brine doesn't give. But the footage doesn't prepare you for how fast it happens. Researchers, watching the early ROV recordings, expected to see some kind of transition. A moment of distress followed by a gradual decline, the way most toxic exposures work in biology. What they saw instead was a switch, an animal moving normally on one side of the boundary, and then an animal that had lost control of its own body on the other side of it, within a time frame so short that the two states seemed almost simultaneous. There was no visible warning, there was no visible struggle, there was just before and after. But the speed makes sense once you understand what osmosis does at the cellular level. The process doesn't require time to build. It doesn't require repeated exposure or accumulating damage. The moment a cell adapted to one concentration of salt encounters a radically different concentration, the physics begins immediately. Water moving across the membrane at a rate that the cell has no mechanism to resist, because the gradient driving that movement is so extreme that no biological process operates fast enough to compensate. The cell is not being poisoned in the conventional sense. It is being physically collapsed by a pressure differential it was never built to encounter. But the salt is only one layer of what kills. Brine pools are also anoxic. The microbial activity at the sediment boundary strips oxygen from the water, leaving concentrations too low to sustain any aerobic organism. And many pools, particularly in the Gulf of Mexico, carry hydrogen sulfide in quantities that are independently lethal through a completely separate mechanism that has nothing to do with osmosis. An animal entering the brine doesn't face one threat it might theoretically adapt to, given enough evolutionary time. It faces a combination of threats, each arriving through a different biological pathway, all arriving at once. The redundancy is the point. There is no way in and back out. Death in a brine pool is not slow. There is no gradual deterioration, no prolonged struggle. The process that kills is osmosis, and osmosis operates at the speed of chemistry, which is to say, faster than any animal can react. A marine cell adapted to seawater at 3.5% salinity, suddenly surrounded by brine at 30% salinity, loses water through its membrane in milliseconds. The concentration gradient across the cell wall is so extreme that the cell effectively deflates, its proteins unraveling, its structural integrity collapsing before any signal of distress has time to travel to the brain. The salt alone would be enough, but brine pools carry more than salt. The water is also anoxic, stripped of oxygen by the microbial activity that occurs where brine meets sediment. Many pools, particularly in the Gulf of Mexico, are also saturated with methane and hydrogen sulfide at concentrations far beyond what any marine organism can tolerate. Hydrogen sulfide binds to hemoglobin, and once that happens, blood loses its ability to carry oxygen, regardless of how much might theoretically be available elsewhere in the water. An animal entering a brine pool doesn't encounter one threat. It encounters a compound failure of every biological system at once: dehydration at the cellular level, hypoxia from the anoxic water, and chemical poisoning that shuts down circulation simultaneously. The redundancy of the killing is what makes it so absolute. Therefore, nothing survives full immersion. Not fish, not crabs, not eels, not organisms that live and thrive in every other part of the deep ocean. The brine doesn't discriminate, and it doesn't give second chances. The boundary between brine and seawater is invisible. There is no color change, no obvious warning, just water and then suddenly different water. By the time the difference registers biologically, the damage is already done. So the killing is instant. There is no recovery, no escape once the boundary is crossed. No biological system fast enough to compensate for what the brine does to a living cell. But that raises something researchers didn't fully anticipate when they first reviewed the footage. If nothing survives entering the brine, and if nothing from outside the brine will cross the boundary to consume what's inside, then the bodies don't go anywhere. They stay exactly where they fell, and some of them have been there far longer than anyone initially assumed. One of the first things researchers noticed reviewing footage from early brine pool dives was the animals, not the living ones at the perimeter. Those came later, once the cameras had panned out and the full extent of the ecosystem became visible. What researchers noticed first were the dead ones. Scattered across the lake bed, visible in the ROV lights, were the bodies of creatures that had entered the pool and not come back out. Crabs, fish, eels, each one lying exactly where it had come to rest, in the position it had assumed at the moment it stopped moving. In the open ocean, a dead animal disappears fast. Scavengers find it within hours, sometimes minutes. The deep sea has its own recycling system, efficient and thorough, built on organisms whose entire existence is organized around locating and consuming organic material before something else does. A whale that sinks to the seafloor will support an entire temporary ecosystem for years, stripped down to bone by a succession of scavengers working in order of size and speed. Nothing goes to waste. Nothing stays intact for long. But the bodies in a brine pool are different. Some of them, based on the state of preservation and the accumulation of sediment around them, have been there for years. The crabs are still whole. The fish still have their eyes, and the eels are still coiled in the exact position they held at the moment they stopped moving, as though they simply lay down on the bottom of the pool and never got back up. Nothing has touched them, nothing has scattered them. The question is, why? And the answer, when it comes, reveals something about the chemistry of brine pools that goes further than just the killing. But the preservation raises an immediate problem. If nothing enters the brine and survives, and if the brine is lethal to every organism that crosses the boundary, including the scavengers and bacteria that would normally consume a dead body, then the lake bed should be accumulating corpses indefinitely. Every animal that has ever made the fatal mistake of crossing that line is still there, in the order it arrived, going back as far as the pool has existed. But that raises a question that researchers found harder to answer than expected. How old is the oldest body down there? The chemistry preserves organic material, and the barite casing slows decomposition to a near halt, but not completely. Given enough time, even in anoxic hypersaline conditions, organic material will eventually break down. The question is how much time? Researchers studying the Neom pools found crabs intact after 8 years, which confirmed the preservation mechanism was working. But 8 years is a short window. The pools themselves are far older than 8 years, which means the lake bed should contain material going back much further. And the further back you go, the harder it becomes to date what you find. But the dating problem points to a question nobody had fully anticipated before the Neom discovery. If brine pools can sit undiscovered in well-surveyed water for decades, and if the preservation inside them is as complete as the evidence suggests, then the oldest bodies on those lake beds may represent species, behaviors, or population patterns that nobody has yet catalogued. Organisms from regions of the ocean that researchers had not prioritized studying, preserved in the exact condition they were in at the moment they crossed the boundary, potentially decades or centuries before any scientist thought to look there. The age of the oldest preserved body in any given pool is unknown. The species it might belong to is unknown. And the only way to find out is to go back down. What the brine destroys, it also keeps. That is the central fact of the graveyard, and it is what makes the seafloor of a brine pool unlike almost any other environment on Earth. Decomposition requires oxygen. It requires bacteria that can metabolize organic matter. In normal seawater, a dead fish disappears within days. Scavengers strip it, bacteria break it down, the skeleton disperses. In a brine pool, none of that happens. The water is anoxic, so aerobic bacteria cannot survive there. The salinity is so extreme that even the microbes adapted to deep-sea sediment cannot function inside the pool. The scavengers that patrol the pool's edges will not cross the boundary. Therefore, whatever falls into the brine stays in the brine, exactly as it was. The chemical environment goes further than simply preventing decay. Barium and sulfate dissolved in the brine react with organic material, forming a barite crust, a mineral shell that encases the body and locks it in place. Therefore, the corpses don't just sit on the lake bed. They are progressively entombed in it, preserved at the molecular level through a process that requires no intervention, no cold storage, no controlled environment. Just the chemistry of the brine, working slowly and completely in the dark. At the Neom pools, crabs were found still intact after 8 years. At other pools, researchers estimate some of the preserved bodies may be decades old, and the brine gives no indication of releasing them anytime soon. What this means is that a brine pool lakebed functions as an involuntary archive, not of specimens that were collected and catalogued and placed there by any intent, but of everything that crossed the wrong line at the wrong moment, preserved by the same chemistry that killed them in the order they arrived, going back further than anyone has yet been able to date. The brine doesn't select. It keeps everything indiscriminately with no mechanism to stop. so the brine preserves what it kills. But in 2016, the footage that most of the world eventually saw was not from the Red Sea. It came from a pool in the Gulf of Mexico, roughly a day's boat ride from New Orleans, a pool with a name that fits what happens inside it. The Nautilus expedition filmed it in high definition, and what the cameras captured at the boundary lasted 4 seconds and ended only one way. By 2016, brine pools were not unknown to science. The first documented reference to anomalous hypersaline water at the bottom of the Red Sea dated back to 1964. Several pools had been surveyed in the Gulf of Mexico and the Mediterranean over the following decades. The basic physics was understood: the density differential, the boundary maintenance, the lethality to marine life. Researchers knew what brine pools were and roughly how they formed, but knowing something exists and being able to show it to the world are different things. The footage from early brine pool dives was limited, grainy, short, not always clearly explained, not widely distributed outside specialist journals and conference presentations. The general public had no particular awareness of brine pools as a phenomenon. They existed in the literature, in the data, in the quiet accumulation of scientific papers that most people never read. That changed in 2016, when a specific pool in the Gulf of Mexico was explored in detail by the research vessel Nautilus, and the footage its RV Hercules transmitted back to the surface was released publicly. The clip spread across science media, then general media, then the internet at large, shared by oceanographers, science journalists, teachers, and people who had never shown any interest in deep-sea research and found themselves, to their own surprise, unable to stop watching it. The pool had been known to researchers before the Nautilus expedition. Its approximate location was on record. Its chemistry had been sampled. Its existence was documented. But no one had filmed it in high definition, and no one had stayed long enough to watch what happened at the boundary in real time. But the footage showed something that the earlier scientific descriptions of brine pools had not fully conveyed. The pool looked inviting. That was the word that researchers and journalists kept reaching for when they tried to describe the Hercules footage to people who hadn't seen it. Not threatening, not obviously dangerous, not the visual equivalent of a warning sign. The surface of the brine shifted in slow, gentle ripples. The slight warmth of the water created a shimmer at the boundary that, in the right light, looked almost welcoming. Nothing about the image communicated instant death, but the animals at the perimeter knew. That was the detail that made the footage so difficult to look away from. The crabs and fish patrolling the edge were not behaving randomly. They were approaching the boundary, testing it with antennae and fins, getting close enough that a single misstep would have been fatal, and then pulling back. They had learned the line or evolved the sensitivity to detect it, or both. The boundary that was invisible to a camera was apparently detectable to the nervous systems of the animals living next to it. They knew where it was. Most of the time, they respected it. But not always. The Hercules footage captured the exact moment one fish failed to maintain that margin, not swimming deliberately into the brine, but drifting too close while pursuing something. The tip of its tail touching the surface, and the cascade of biological failure that followed, happening faster than the fish could correct for. 4 seconds. That was the full duration of the sequence from contact to stillness. The fish did not fight for long. The brine didn't require it to. The Jacuzzi of Despair is roughly 30 meters in circumference and about 3.5 meters deep. By the standards of brine pools in the Gulf of Mexico, it is not large. What made it famous wasn't its size, it was the footage. In 2016, the research vessel Nautilus deployed its ROV Hercules to a site about 1,000 meters below the surface, roughly a day's boat ride from New Orleans. The footage the ROV transmitted became one of the most widely shared clips in deep-sea research, not because of what it showed scientists, but because of what it showed everyone else. The pool sat in a crater-like depression, its surface slightly warmer than the surrounding water, methane bubbling up gently from beneath, the brine shifting in slow, almost peaceful ripples. From a distance it looked, against all reason, like something you might want to sit in. Around the perimeter, life was dense. Mussel beds covered the sediment in thick, overlapping layers. Tube worms stretched upward from the rock. Amphipods moved in clouds through the water column. Fish patrolled the outer edge in slow, deliberate circuits, hunting the crustaceans that gathered near the bacterial mats coating every surface. The ecosystem around the pool was thriving, concentrated, active, and operating entirely on the chemical energy the pool itself was producing. Therefore, the pool was not a dead zone. It was the engine of the only thriving habitat for hundreds of meters in any direction. But inside the pool, there was only the graveyard. Dozens of dead crabs on the bottom, shells intact, claws still positioned as they had been at the moment of death. A fish here, an eel there, each one preserved exactly where it settled. The footage from Hercules captured one fish in the exact sequence of crossing the boundary, tail brushing the brine surface, a single spasm of the body, sideways drift, and then stillness. The fish sinking slowly through the denser water to join the others below. The moment lasted about 4 seconds. Therefore, what the Jacuzzi of Despair showed was not just what brine pools are. It showed what the boundary actually looks like: invisible, absolute, and surrounded by life that knows, somehow, exactly how close it can go. So the Jacuzzi of Despair showed the boundary the way no prior description had: visible, absolute, and operating in real time. But the footage showed something else too, something that took longer for most viewers to process. The animals at the perimeter were not trying to avoid the pool. They were circling it, returning to it, feeding along its edge with a precision that suggested the boundary was not a threat to them, but a resource. The pool that kills everything inside it appears to be sustaining an entire ecosystem just outside it, and the reason why changes what brine pools actually are. The first assumption most people make about brine pools, after learning what the water does to anything that enters it, is that nothing lives near them. The logic is intuitive. If the pool is instantly lethal, and if the chemicals it contains are toxic at concentration, then the area around a brine pool should be empty, a dead zone, avoided by every organism in the vicinity. The way animals avoid hydrothermal vents that are too hot or volcanic zones that are too acidic. Early brine pool surveys seemed to confirm this. Food is scarce. At those depths, pressure is extreme. And most organisms that live on the deep seafloor are small, slow-moving, and spread thin. A few creatures per square meter across stretches of sediment that can run for hundreds of kilometers without any significant feature. On a first pass, the water column around a brine pool looked like the water column everywhere else at that depth. Dark, quiet, nearly empty. But subsequent expeditions, with better cameras and longer bottom times, began to tell a different story. The perimeter of a brine pool is not empty. It is, by the standards of the deep ocean, extraordinarily crowded. The organisms living there are not scattered or sparse. They are layered, concentrated, stacked one on top of another across every available surface in densities that researchers more commonly associate with shallow water reef systems than with the lightless depths of the ocean floor. What none of the initial surveys had been able to explain was where the energy was coming from. Life requires fuel. Even the most efficient deep-sea organism needs a source of chemical or biological energy to sustain itself. The seafloor at those depths offers very little. Occasional marine snow drifting down from the surface, the bodies of dead organisms, the slow oxidation of sediment. None of that explains the concentrations of life found at brine pool edges, which are too dense and too organized to be accounted for by the background energy available in deep water. But the density of life at the brine pool edge creates a situation that has no clean parallel anywhere else in deep-sea ecology. In most extreme environments, the danger zone and the habitable zone are clearly separated. You are either in the toxic area or you are not, and the organisms that live nearby have evolved sufficient distance from the hazard to function safely. Around a brine pool, that separation doesn't exist. The food is at the boundary. The bacteria that support the entire ecosystem grow at the boundary. The mussels filter feed at the boundary. Everything that makes the edge habitable is also what makes the edge lethal, and the line between the two is measured in centimeters. But what makes the footage from these expeditions genuinely unsettling is that the animals appear to understand this at some level. A crab feeding on a bacterial mat at the pool's edge is not operating with a wide margin of safety. The antenna it extends to probe the surface in front of it is testing chemistry, not distance, detecting whether the water immediately ahead has crossed into a concentration range that will kill it. The crab is doing this continuously, in the dark, at the bottom of the ocean, as a routine part of feeding. The calibration required to live that way, with instant death available at any moment in one specific direction, is not something most ecological models account for. But the cost of getting it wrong is not just individual death. The bodies of animals that miscalculate and cross the boundary don't disappear. They sink into the graveyard on the lake bed and stay there, preserved, adding to the accumulation. The ecosystem at the edge is therefore surrounded on one side by the mechanism that feeds it, and on the other side by a visible record of every creature that the mechanism has already killed. The food and the graveyard share a border, and the animals eating the food are one step away from joining the graveyard. The ecosystem surrounding a brine pool should not logically exist. The deep seafloor is almost entirely barren. Food is scarce, pressure is extreme, and sunlight has never reached these depths. Most of the abyssal plain is effectively a desert, but brine pools are surrounded by some of the densest concentrations of living organisms found anywhere in the deep ocean, and the reason is the same chemistry that makes the brine itself lethal. Methane seeping upward through the sediment around the pool's edge dissolves into the water column in high concentrations. Certain bacteria feed on that methane, converting its chemical energy directly into biological energy. No sunlight anywhere in the process. These bacteria form thick mats across every surface near the pool's shoreline, sometimes several centimeters deep, coating rock and sediment in layers dense enough to be visible from ROV cameras. That microbial foundation is what everything else is built on. Mussels colonize the mats in large numbers, hosting the methane-feeding bacteria in their gills in an exchange that benefits both. The bacteria get a stable surface. The mussel absorbs the organic compounds the bacteria produce, Crabs feed on the mussels and on the mats directly. Shrimp and worms pick through the sediment. Fish patrol the outer boundary, hunting whatever moves. Therefore, the brine pool, which produces nothing biological itself, feeds an entire layered ecosystem through the chemicals it releases at its edges. Chemicals that would kill any of those organisms if they crossed 2 meters to the left. The animals living here have no other option. This is where the food is. Therefore, they stay close to the boundary, close enough to feed, close enough that a single miscalculation means crossing into the brine. The footage from multiple expeditions shows the same behavior. Animals at the edge, constantly testing, occasionally getting it wrong. The ecosystem is not peaceful. It is a permanent negotiation between proximity and death, and the negotiation never ends. So, the ecosystem at the edge runs on poison. The same chemicals that make the brine lethal are feeding every organism that lives beside it through a metabolic process that needs no sunlight and no oxygen to function. But the researchers who began studying those organisms closely noticed something that went beyond ecology. The chemistry those microbes were running on, the specific combination of compounds they were extracting energy from, matched conditions that Earth's oceans had not seen at the surface for nearly 3 billion years. What lives at the edge of a brine pool today may be operating by the same rules as the first life that ever existed on this planet. The microbes living at brine pool edges are not well understood. Scientists have been studying them since the first brine pool surveys produced samples, but the conditions that make brine pools so extreme also make them difficult to study systematically. Sending equipment to 1,000 or 2,000 meters depth, collecting samples without contaminating them, and returning those samples to the surface without the change in pressure and temperature destroying the organisms in transit. Each of those steps introduces uncertainty. The microbes that thrive in those conditions are not the microbes that thrive in a surface laboratory, and the gap between the two environments is large enough that studying one tells you only so much about the other. What the samples have consistently shown, though, is that the organisms living at brine pool edges are operating by rules that most life on Earth does not follow. They don't use photosynthesis. They don't require sunlight at any point in their metabolism. They function in water that would kill almost every other organism on the planet at pressures that would rupture most cell membranes, in chemical conditions that most biological systems would classify as toxic by any measure. And yet they are alive, reproducing, metabolizing, processing energy, building new cells. The question that started bothering researchers was not just how these organisms survive in such extreme conditions. It was whether the conditions themselves were familiar from somewhere. Whether brine pool chemistry, in its combination of specific elements and specific absences, matched conditions that had existed before, not just in some other part of the modern ocean, but at a much earlier point in the history of the planet entirely. Conditions that life had not merely adapted to, but, in a meaningful sense, originated in. But matching the chemistry of a brine pool to the chemistry of the Archean Ocean is not the same as proving that life originated in conditions like these. The overlap is suggestive. It is striking enough that researchers take it seriously. But correlation between a modern environment and an ancient one doesn't establish causation. Doesn't prove that brine pool chemistry is where life actually started, rather than simply being one of many environments where life could theoretically have started if it had been present. The question of origin requires more than a matching set of conditions. But the microbes living at brine pool edges do something that makes the connection harder to dismiss. they are not just surviving in conditions that resemble the Archean Ocean. They are using a metabolic strategy, deriving energy from chemical gradients rather than from sunlight, that predates photosynthesis in the evolutionary record. The organisms that first developed photosynthesis, roughly 3.5 billion years ago, descended from something that didn't use it. Whatever that something was, it ran on chemistry. And the chemistry it ran on was not unlike the chemistry available at a brine pool edge. Hydrogen sulfide, methane, iron compounds, all of it present in the right concentrations, all of it accessible without sunlight. But the deepest problem is that we cannot go back. The Archean Ocean is gone. The conditions that existed 2.7 billion years ago cannot be directly observed, cannot be sampled, cannot be replicated in a laboratory with any confidence that the replication is accurate. The best available proxy, the closest living environment to what those ancient oceans may have looked like, is a brine pool, which means that the organisms living at brine pool edges are not just scientifically interesting in their own right. They may be the only direct window into a period that shaped everything that came after it, and that window is accessible only by sending an ROV to depths where pressure would kill a human instantly. The conditions inside a brine pool match what geochemical models predict for Earth's oceans roughly 2.7 billion years ago, before cyanobacteria began producing oxygen in significant quantities. No free oxygen, methane and carbon dioxide in the water, extreme salinity, geothermal heat rising from below, hydrogen sulfide, and iron-rich chemistry throughout. The modern brine pool is not a perfect replica of the Archean Ocean, but the overlap is close enough that scientists treat it as the most accessible natural laboratory currently available for studying that period. The extranophile microbes living at the edges of brine pools, and in some cases within the outermost layers of the brine itself, derive energy entirely from chemical reactions, with no sunlight involved at any point. That metabolic strategy is considered by many researchers to be the most likely mechanism of the earliest life on Earth, because it requires only the right chemical gradients, and brine pool edges produce those gradients continuously. Therefore, the organisms living at the boundary of a brine pool today may be metabolically closer to the first life on Earth than almost anything else currently alive. Cyanobacteria, which eventually produced enough oxygen to transform the atmosphere in the Great Oxidation Event, descended from ancestors operating through that same basic chemical logic, extracting energy from the environment in conditions that would be lethal to everything alive today. Therefore, brine pools are not just extreme environments at the bottom of the ocean. They are, in a narrow but meaningful sense, a preserved echo of the world that produced every living thing on the planet. Studying what survives in them is the closest thing currently available to observing the conditions of life's origin directly, not through a model, not through a reconstruction, but through chemistry that is still running, right now, miles below the surface. So brine pools may be the closest available window into a period of Earth's history that produced every living thing on the planet, conditions that no laboratory can replicate and no other environment currently preserves. But in 2013, a deep-sea scientist sent a lander to the bottom of the Pacific with a straightforward task: measure how fast oxygen depletes at 5,000 meters depth. The model was clear. The expectation was settled. What the instruments returned instead was data so far outside the predicted range that the team spent years recalibrating their equipment before they would allow themselves to believe it was real. In 2013, Andrew Sweetman was not studying brine pools. His focus was the Clarion-Clipperton Zone, a vast stretch of the Pacific Ocean floor between Hawaii and Mexico, roughly the size of the continental United States, that had drawn scientific attention not for its biology, but for its geology. The seafloor of the Clarion-Clipperton Zone is covered in polymetallic nodules, dense, dark mineral formations that have been accumulating on the seafloor for millions of years through a process that is still not entirely understood. The nodules are commercially interesting, containing concentrations of valuable minerals that mining companies have been studying for decades. And the scientific community wanted to understand what any extraction operation might do to the organisms living there before it began. Sweetman's role was to establish a baseline. Before you can measure the impact of an industrial disturbance on a deep-sea ecosystem, you need to know the state of that ecosystem before any disturbance occurs. Oxygen levels are a fundamental metric. They tell you how much biological activity is happening, how fast organic matter is being consumed, how the system breathes. His team deployed an automated seafloor lander, lowering it to 5,000 meters and letting it sit on the bottom, pulling water samples into a sealed chamber and measuring their oxygen content over time. The expectation was straightforward. Oxygen enters the ocean at the surface, dissolved by contact with the atmosphere, and produced by photosynthetic organisms in the sunlit upper layers. As that oxygen-bearing water circulates downward, marine life consumes it, so oxygen levels should decline with depth. More consumption at every layer, less and less remaining by the time you reach the sea floor 5 kilometers down. That is the model that has governed deep ocean chemistry research for decades. It is the model that every instrument deployed in deep water has been calibrated against, and the model that every oxygen budget calculation for the global ocean has been built on. The lander began returning data. Sweetman's team looked at the numbers, then they checked the instruments, but the instruments were not broken. The team recalibrated them. The readings held, and then they recalibrated again, and the readings held again. And the pattern was consistent enough across multiple deployments and multiple locations within the zone. That instrument error stopped being a viable explanation. Something in the data was real. Oxygen was present in the deep water at concentrations that the standard model said should not be there, not slightly elevated, not within the margin of measurement error, but meaningfully higher than the model predicted, in some areas exceeding the oxygen concentration at the surface above. But the team sat on the data for years before publishing, not because they doubted the measurements, but because the implications were significant enough that they wanted no avenue of challenge left open, a finding that contradicts the foundational model of deep ocean oxygen chemistry, a model that has been calibrated against decades of consistent data from instruments deployed all over the world, is not the kind of finding you release on the basis of one expedition, or two, or three. Every time they ran the numbers again and the anomaly persisted, it became harder to explain away. And every explanation they tested for how the oxygen might have gotten there through conventional means failed to account for the scale of what the instruments were recording. But the most uncomfortable part of the anomaly is what it implies about the model itself. Every study of deep ocean life that has been conducted under the assumption that oxygen only enters the system from the surface, every calculation of how fast deep-sea ecosystems are consuming available oxygen, every projection of how deep ocean chemistry will change over time was built on a baseline that may be wrong. The anomaly is not just a strange local observation in one stretch of the Pacific. If the mechanism producing the oxygen operates wherever the right geology exists, then the standard model of deep ocean chemistry has a gap in it that nobody knew was there. Andrew Sweetman and his team ran the measurements for years before publishing. The data was too strange to release without exhaustive verification. Instruments were recalibrated multiple times. Independent teams checked the methodology. The results held every time. At 5,000 meters depth in the Clarion-Clipperton zone of the Pacific Ocean, oxygen levels were not declining with depth the way every model of ocean chemistry predicted. In some areas, the water near the seafloor contained more dissolved oxygen than the water at the surface above it. Therefore, something on the seafloor was generating oxygen in complete darkness, under crushing pressure, with no photosynthesis occurring anywhere nearby. One hypothesis centers on polymetallic nodules, potato-sized mineral formations that cover large sections of the deep Pacific seafloor. These nodules contain manganese, cobalt, nickel, and iron in concentrations that create electrochemical potential across their surfaces. The hypothesis is that the nodules may be producing a weak electrical current through geochemical reactions, and that current may be splitting water molecules into hydrogen and oxygen, a process called electrolysis, happening spontaneously on the seafloor with no organism and no sunlight driving it. If that mechanism holds, the consequences for deep-sea chemistry are immediate and measurable. The oxygen anomaly means that every prior calculation of how much oxygen exists in the deep ocean and how fast it is being depleted may be wrong. Scientists have been modeling deep ocean oxygen budgets for decades under the assumption that oxygen only enters the system from the surface. If the seafloor is producing it independently, those models need to be rebuilt from the beginning. The deep ocean is not running out of oxygen on the timeline anyone predicted. And the reason why is something that, until 2013, nobody knew to look for. So the oxygen model for the deep ocean may be wrong, built on a baseline that didn't account for a process the seafloor has apparently been running the whole time, undetected in the dark. But the implications of that anomaly don't stop at the ocean floor. Several moons in this solar system are now known to contain liquid saltwater oceans beneath frozen surfaces, oceans with no sunlight, no photosynthesis, and chemistry that may not be entirely unlike what sits at the bottom of the Red Sea. If mineral chemistry can generate oxygen without sunlight, and if life can run on chemical gradients alone, then the barrier that ruled those oceans out as candidates for life may not be a barrier at all. The question of life beyond Earth is not a new one. It has been asked in various forms for centuries, and the scientific version of the question, what conditions are required for life to exist? And do those conditions exist elsewhere in the solar system? Has been active since the space age began. For most of that time, the working assumption was that life requires liquid water, a source of energy, and the right chemistry. The energy source, in every model built on the biology scientists could actually study, was sunlight. Photosynthesis drives the base of almost every food chain on Earth's surface. Remove sunlight, and the chain collapses. That assumption shaped where scientists looked. Mars received the most attention because it has evidence of ancient liquid water on its surface and sits within a distance from the Sun, where liquid water is at least intermittently possible. The outer solar system, Jupiter, Saturn, their moons, was considered largely irrelevant for life, too far from the Sun for any photosynthesis-dependent chemistry to function. The moons of Jupiter and Saturn are frozen, dark, and receive a fraction of the solar energy that reaches Earth. By the photosynthesis-dependent model, they were dead. Then, in the late 1970s and 1980s, scientists studying hydrothermal vents on Earth's ocean floor found something that didn't fit the model. Entire ecosystems were living in complete darkness, at crushing depths, sustained by chemical energy from the vents rather than by sunlight from above. The base of those food chains was not photosynthesis. It was chemosynthesis, bacteria extracting energy directly from chemical reactions involving hydrogen sulfide and other compounds. The discovery didn't immediately change the search for extraterrestrial life, but it introduced a possibility that hadn't existed before. Life without sunlight, operating entirely on chemistry. Spacecraft began returning data from the outer solar system that made the possibility harder to ignore. But the chemosynthesis discovery, on its own, wasn't enough to reopen the question of life in the outer solar system. One ecosystem in the dark near a hydrothermal vent powered by hydrogen sulfide. That was extraordinary, but it was local. It required specific geological activity to produce the chemical gradients the bacteria needed. Not every ocean floor has active hydrothermal vents, and the oceans of Europa and Enceladus had not confirmed to have them either. Current evidence suggests they might, but the evidence is indirect, derived from remote sensing and spacecraft flybys, not from anything we have actually placed on or near those ocean floors. But the oxygen anomaly changes the calculation in a specific way. Hydrothermal vents require active geology, a heat source, a specific set of rock-water interactions, conditions that may or may not be present. Mineral electrolysis, if it proves to be the mechanism behind Sweetman's findings, requires none of that. It requires only the right minerals on the seafloor and water in contact with them. Polymetallic nodules form through extremely slow geochemical processes that are not unique to Earth. Similar mineral formation processes have been identified on other bodies in the solar system. If oxygen can be produced by mineral chemistry alone, then the geological threshold for a habitable ocean is lower than anyone calculated when the search for extraterrestrial life was first being designed. But the harder problem is that we are not close to being able to test any of this. Europa and Enceladus are not accessible in any meaningful sense with current technology. No lander, no submersible, no instrument package exists that could reach their oceans and return data on what is living in them, if anything is. The evidence that makes those oceans interesting as candidates for life is also the evidence that makes them almost impossible to study directly. They are covered in ice. Their oceans are sealed. Whatever is happening in the water beneath that ice is happening in complete isolation from anything we can reach. And the gap between knowing that something might be there and being able to confirm it is measured not in years, but in the slow timeline of spacecraft development, mission planning, and the decades those missions take to arrive. Europa's ocean is estimated to be between 60 and 150 kilometers deep. It sits beneath a surface of ice several kilometers thick, with no sunlight reaching it and no photosynthesis possible at any depth. Enceladus has an ocean confirmed to contain organic molecules, hydrothermal activity, and saltwater chemistry. The Cassini spacecraft detected plumes of water vapor erupting from its surface in 2005, and analysis of those plumes showed the presence of molecular hydrogen, a byproduct of the kind of water-rock chemical reactions that occur at hydrothermal vents on Earth's ocean floor. Those are the same kinds of reactions that sustain the chemosynthetic ecosystems around brine pool edges. The same basic chemistry that keeps the bacterial mats and mussel beds alive at the jacuzzi of despair may be occurring in the ocean of Enceladus right now, 1.2 billion kilometers from Earth. The conditions are not identical. They are never identical between two different bodies in the solar system. But the relevant question is not whether conditions are identical. It is whether the chemical gradients required for life to operate without sunlight are present. And in both Europa and Enceladus, current evidence suggests they may be. Less of Earth's deep seafloor has been mapped than the surface of Mars. The 4 neon brine pools sat undiscovered in the Red Sea, not in some remote trench in the middle of the Pacific, but within sight of a populated coastline until someone happened to point a sonar at the right patch of water in 2020. No instrument currently exists near Europa or Enceladus capable of detecting what might be living there. What has accumulated, though, is a growing catalogue of environments on this planet where life operates entirely on chemistry, in permanent darkness, under conditions that nothing alive today would voluntarily enter. And that catalogue gets longer every time someone points a camera at the seafloor and finds something that, by every prior assumption, should not be there.